Welcome to Unsupervised Thinking, a podcast about neuroscience, artificial intelligence, and science more broadly. We're a group of PhD students studying computational neuroscience. I'm Grace.
I'm Josh.
I'm Conor.
And I'm Nancy.
Nancy is our special guest today because our topic is neural oscillations, and that is something that she studies and none of the rest of us do. So she's the resident expert for today. So I guess we should start by explaining what we mean by neural oscillations, and I think it's a term that people are probably familiar with in some way, like even just in natural language, people talk about brain waves and that kind of thing. So there's probably a lot of ideas out there about what neural oscillations are. So we should start by kind of laying out what scientists in the field study when they study neural oscillations.
So Nancy, maybe you could get us started with that and describe those, and then also how you measure oscillations.
Mm-hmm. So besides spikes that are generated by neurons, there's a lot of other electrical changes in the brain, and they sometimes happen in rhythms, and that's why we call them oscillations. And we can simply record them by putting an electrode anywhere inside the brain. Even you can record them in your skull because they're that powerful, and you can listen to them. It's like the echoes pretty much. So that's what we know we refer to as oscillations or neural oscillations.
So what do we know about how these things are generated?
So they're a mixture of spikes and subthreshold activity, like what we refer to as excitatory postsynaptic responses or inhibitory postsynaptic responses, and also just ongoing currents that are occurring constantly. So what we will call not synaptic activity, extrasynaptic activity.
Yeah, so neurons fire spikes, which are these, kind of sharp increases in membrane potential that kind of allow them to communicate via synapses with other neurons. The way that those spikes are generated is by some activity in, flowing currents, basically literally ions flow across the membrane. So there's a whole lot of currents going on all the time in the brain across the membranes of cells. And then, so Nancy referred to, subthreshold activity. That's like the changes in the membrane voltage, so the voltage across the body of the cell that are below the spiking threshold. So changes that happen when there is no spiking occurring.
Right, and maybe it's worth mentioning that neuroscientists care a lot about spikes, and we give it like very strong meaning because when an action potential occurs, you have the release of neurotransmitters, and that's what we call the message. The neurotransmitter is then going to affect the neuron sitting next to it. Now, with this subthreshold activity, we don't really know what happens. There's no release of neurotransmitters per se.
And people might think of oscillations that they record extracellularly as being maybe the sum over the subthreshold activity of many neurons.
Exactly.
Kind of ill-defined, possibly number of neurons.
Right.
And so, as Nancy said, you can measure these oscillations by sticking an electrode in the brain, and that signal is usually called the local field potential if you're actually sticking an electrode inside of the brain and capturing something that's happening across maybe hundreds of neurons. And then if you're recording from the skull, those oscillations, when you measure that, is usually called an EEG.
And there's some intermediate-sized recordings where people put grids of surface electrodes on the surface of the brain, touching the brain inside the skull, but not penetrating into the brain. And this is generally known as like ECOG.
So I think the idea of an oscillation, right, in someone's mind, if you think of like something oscillating, you would expect that if you say a quantity is oscillating, if you plot that quantity as a function of time, you would literally see something going up and down, right? Mm-hmm. But
this is not exactly what
this is not what people are often talking about when they talk about like oscillations in neural signals, right? Because there are examples where that's really true, but oftentimes it's not the case that if you just looked at the signal that's recorded on the electrode that you would actually see an obvious oscillation.
Right. They're not there by eye.
Right, that's true. And usually they're evoked by something. I mean, if you're recording in a behaving animal, if you're in the hippocampus, for example, the animal starts moving and locomotion triggers this very, very defined oscillatory activity. Same in the cortex as well. But if sometimes just at baseline there's no oscillating activity. So most of the time it's evoked, I would say.
So if you can't see it by eye, then how do we actually measure these things?
Right, so maybe something worth mentioning is that in the local field potential, there's actually many frequencies over on top of each other, and we use mathematical methods to extract different frequencies and filter them, if you may.
So what you're effectively getting is the power of different frequency components, like how much of low frequency component is there in this local field potential, or how much of a high frequency component of this. And neuroscientists have named these different bands of frequencies different things. So like, what are some of the dominant?
So one of the very popular ones are theta, which is low frequencies from 4 to 12 hertz. That means 4 to 12 cycles in a second, hertz. And another popular one is gamma, which is really anything higher than 30 hertz.
Cool.
Yeah, and it's true. We measure specifically like the amplitude of that signal that is going up and down. And the idea is that in different behavioral states, those amplitudes of the given frequencies are changing. So perhaps there's a meaning of these oscillations.
So I think historically kind of the study of oscillations came when they realized that during sleep there were slow oscillations that you can measure with EEG. And then during wakefulness, it's considered not really oscillating. During epilepsy, epileptic seizures, there's huge oscillations. So that was kind of the initial sense that you could correlate, when there's oscillations, you're kind of not processing information or in a not alert state, and when there's not oscillations, maybe you are. But then obviously people have gone further and looked at these oscillations inside the brain and found kind of more detailed patterns that they try to correlate with behavior.
At this point, maybe having talked a little bit about this as background, we could have Nancy describe a little bit of how she uses these tools in her research.
Okay. So one of the main motivators for us to use what we call local field potentials is that they can easily be translated to human research because, as Grace mentioned, you can record these oscillations from the skull, and they're known as EEGs. So the idea is that we want to use local field potentials as markers of functional connectivity. And by that, we mean that two brain regions are engaged in a given behavioral state. In our case, the behavior I care about is anxiety or innate fears. So an animal is naturally afraid of specific stimuli because of evolution. In our case, we use these mazes that have very bright spaces that rodents dislike. So we've previously reported that the ventral hippocampus and the prefrontal cortex become synchronized, which means that the oscillations from the ventral hippocampus and the prefrontal cortex look more similar to each other in amplitude and in frequency. So then we are essentially saying these two brain regions are functionally connected. And so essentially we're using the local field potential more as a way to ask, are these two brain regions engaged together, yes or no? It's easier than just recording spikes because recording a lot of spikes in multiple brain regions is very, very challenging. And even if you do so, you're probably assaying less than 1% of the neurons in that brain region. So the local field potential offers you this summation of synaptic activity that can get around the limitations of recording single units, as we know them, referring to neurons.
So when you record these signals, you kind of think of it as some kind of heuristic or aggregate bulk signal for like a whole ROI, or region of interest in the brain.
Yeah.
And how do you think about what kinds of statements you could make about the sort of causal interactions, just kind of at a high level? I mean, what kind of statements do you think it's possible to make about the interactions between these brain regions, given these recordings? How strong of a statement would you feel comfortable personally endorsing based on this kind of science?
Well, it's similar to what fMRI people do. You look at the change. So you're recording some baseline condition, and then you're recording your behavior of interest. At least that's how we do it. The problem is that these oscillations are occurring all over the brain. You do have volume conduction happening, which means that the signal I record in the ventral hippocampus could partially be getting some noise from regions around it. So what we're comfortably talking about is changes. So we can see that the amplitude increases from the baseline condition to when this stimulus is presented, for example.
So I don't want to push you too hard. I just want to get a clarification of, right? So in your mind, what are the kind of, how strong of a statement can you make though from this? Like when you would you say like what one brain region is causing or transferring information to another brain region, or would you say that's beyond the scope of what I can probe with?
That's definitely beyond the scope, but we can talk about how much a signal in one brain region is influencing the other. Causing is definitely, there's no way. I mean, the brain is completely interconnected. So
what would be the distinction to you there? What do you mean by influencing?
Influencing means that, let's say in my case, the prefrontal cortex is the region that receives projection from the hippocampus. And the prefrontal cortex seems to follow the oscillation from the ventral hippocampus. And that doesn't mean that the ventral hippocampus is causing the oscillation in the prefrontal cortex, but it's influencing. So partially, some of that signal on the PFC is due to the signal on the hippocampus. That's as much as we would say.
Is it possible to ever, I mean, this kind of standard, right, where you would want to manipulate the thing that you think causes something else?
So that's exactly what my PhD project is about. So now that we have opgenetics, which is a way to silence or excite given parts of the brain with very precise temporal and spatial resolution, the idea is that we've previously reported that these two brain regions their oscillations become more similar to each other when the animal is anxious, but we don't really know if that has any meaning. So we thought, well, let's silence, let's control, let's manipulate this projection and see if that coordinated activity between the hippocampus and the prefrontal cortex decreases. You will expect that it should, right? Because if your technique is working, you're silencing that projection. I mean that those neurons that project to the prefrontal cortex. And we do see that the changes in behavior and the changes in synchrony go together, suggesting that at least it wasn't a byproduct of something else that was happening during the behavior. And when you manipulate and you silence that connection, you do see decreased phase locking, which means like the neurons in the PFC are not as rhythmically synced to the hippocampus.
Are you also changing firing rates and spiking?
Right, so what's interesting is like overall firing rate doesn't change, but evoked firing rate changes. So if I look at firing rate in a specific task of the behavior, it does change in a specific part of the task, but overall it doesn't. So it's maybe what we're thinking is we're disorganizing the spikes. Like maybe the spikes that would occur in a given part are occurring in another part because the total amount of spikes is not changing. I mean, if you think about it, I'm only decreasing one of the many inputs and I'm recording cells that could be receiving hippocampal input or not. So in vivo, it's not the best assessment. I'm sure there's cells that are significantly decreasing firing rate, but maybe in vitro, you could definitely ask the question, can I control for the amount of spikes within one cell and still disorganize this phase, the spiking to the LFP?
Just a tiny brief aside, terminology, in vivo versus in vitro. In vivo is doing experiments in living animals. In vitro is doing experiments with tissue that might be alive, but in like a dish, separated from the living animal.
So if you do something in vitro, is there a strong LFP, or can you only study LFP in vivo?
No, you get strong LFP, definitely. Now, the LFP is I'm not an expert in vitro, so I believe that the LFP is not as strong in terms of the oscillation. The rhythmic activity is not very strong, so people usually induce it pharmacologically.
Okay, so maybe we can move on to the paper and kind of get a sense of what tools people generally think they can use with respect to oscillations and what kind of science can be done. And then we can return to this and kind of discuss more broadly to the extent to which we endorse or have qualms with some of these usages.
Right, so we read a paper by Bujaki and Schomburg, who are researchers at NYU. It's called What Does Gamma Coherence Tell Us About Interregional Neural Communication? So this paper is kind of based in an idea that people have that if you have coherence between two areas, that is, you have two areas that are oscillating the same way, that can facilitate communication between those areas. And this paper just goes into perhaps some of the things that people should keep in mind when they're doing those kinds of studies, because sometimes it can be difficult to study oscillations, as we've talked about. It's sometimes not clear what exactly you're studying when you're studying oscillations, and if you want to say something really strongly, you need to know exactly what you're measuring and what can be said with that kind of signal.
So can I ask a question, just kind of stupid? If you have some signal, say the voltage recorded from the end of an electrode in the brain somewhere, you can do like a Fourier transform of it. So Fourier transform splits a signal up into a bunch of sine waves that have different frequencies. And you can kind of see how much of the signal is contained at all the different frequencies, up to some maximum frequency. So to me, like being a bit mathematical about it, I wouldn't feel comfortable looking at a signal and calling it oscillatory unless there was like some kind of band pass peak in the Fourier spectrum somewhere. Or at least if I could like do a local Fourier transform and find strong peaks in that might, say, change over time in their amplitude and so on.
And this would mean that by eye, when you look at the signal...
When you look at the power spectrum?
Well, no, but this would mean that by eye, when you look at the signal, you would see
Catch it.
Oscillatory activity by eye.
Depending on, like, you could be able to... You might talk about, is there a peak or not that's bandpass in some statistical way. You could test the significance of that. It might be embedded in a lot of noise. It might be hard to see it clearly by eye.
You're just questioning, taking it for granted just from the power spectrum itself.
Like if you have like some kind of, power spectrum that decays like 1 over the frequency.
1 over f.
Or
then
something,
there's nothing.
or whatever, then it's kind of hard, we know that those kinds of spectra can arise from various kinds of noise processes. So it's like, my question is basically about, because I actually don't know the answer to this. There's all these different oscillations people talk about. I know that the theta oscillation in the hippocampus is like, you look at that, you see a big peak. It's really clear. So it's like that exists in an obvious way.
Right.
And many of these exist in like an obvious way.
Yeah. Yeah, so people that do this correctly, they study an oscillation that is perceived as a peak in a power spectrum. Otherwise there's nothing, right? Because for mathematical reasons that I do not understand, if you take noise, like if I generate an LFP that's like just simply white noise, and you do a power spectrum on that, and there's nothing really oscillating on it, there's nothing rhythmic to it, you'll see a power spectrum. You can look at the frequency components on anything, and it'll look like 1 over f, so it'll look like these exponentially decaying things.
So just, for sort of background context, right, if you generate just a random noise signal and look at the spectral content of that, depending on the way you generated that random noise, you'll expect to see a different frequency spectrum, power as a function of frequency, and there'll be different amounts of those the different frequencies.
Right, you get some random values.
You'll have, so white, when you generate something that looks like white noise, so like random, at each time you have like a random value drawn from some distribution, that tends to mean statistically that there is frequency content at all frequencies, right? This is the meaning of white noise, is that there's some
all over
there's
the
some
place,
power
right?
at all the frequencies,
and it's uniformly.
It's often considered it's often considered to be strictly uniform, though sometimes there are slight variants on that. People still
call it uniform. Typically, white noise means that there is actually
power at all frequencies.
It means there's power at all frequencies.
Anyway, so if you take a randomly a signal that you make up that's not recorded from the brain, you can get amplitude signal values like as if there were oscillations on it, because the Fourier transformation will look for essentially it will look for the different frequencies even if you cannot see them by eye.
So the question is, how do you determine that the thing you recorded from a brain actually is a significant oscillation?
Right, so it should be a peak that looks bigger than 1 over f, which is what you will get with this random signal. Now, I'm not sure if there's specific statistics that people use, but the way that people do it is that they're recording, they see this particular peak in, let's say, I don't know, between 6 and 9 hertz in a specific behavior, and then they ask questions about, does this have a meaning? Is it somehow linked to the animal's behavior on a given thing, and can I manipulate the animal's behavior and will that thing change? So it's more like it's like blindly you record and you see what oscillations you see in this paradigm, rather than just not checking if it actually exists. Because at the beginning, people thought that this was very uniform across the brain, but it's brain region dependent and behavior dependent. So
I think it's actually a slightly tricky issue, right? Because, for example, when you then talk about coherence between signals, so there's probably like a few different definitions people use, but let's just say we have two signals, one neuron, one LFP recording one brain area and LFP recording another brain area. And suppose they're kind of like, suppose the spectra of these two signals are like basically flat. They have frequency everywhere, right? Mm-hmm. Now suppose we filter each of them in some frequency band. We extract the, say, 50 to 55 hertz components of both of those, right? You could then ask questions about, is there meaningful relationships just between the frequency, those two filtered signals? You could ask, like, are they always in phase? Is one always slightly preceding another? Mm-hmm. And I think a lot of these ideas that we'll get to about coherence, communication, if they're plausible, that kind of scenario, they could still exist. But then, in terms of what we've just been talking about, you might still look at each signal and say, it doesn't itself have some strong gamma oscillation, right? But it could be that there's I don't know how this would arise, but it could be that there are at certain frequencies, the signals are related, and other frequencies they're not.
So absolutely.
These things kind of all are possible, and I think it's not totally clear what's interesting and what's relevant. So.
Yeah, two signals could still be influencing each other if they're not their LFPs are not reflecting the same frequency ranges or the same oscillation in the same frequency.
But again, for context, there is a concrete mathematical definition of coherence used by signal processing people, which is effectively it's a cross power spectrum that's normalized by the individual power spectrum. Mm-hmm. So it's related to the cross correlation function. Mm-hmm. So kind of, if you would expect to see that kind of, and it's as a function of frequency. So basically, it's at each frequency how strong is how strong are two signals cross correlated. Yeah.
But they don't need to each be oscillating, basically, is my point, for you to have.
You'll get all the frequencies just because that's...
Well, so if within a given frequency band, the signals are not varying together, then you won't expect to see something.
Yeah.
So being in phase and having amplitude that kind of covaries together is going to...
What I'm saying is that
increase the coherence.
Two signals could have coherence in some frequency band without each of them actually looking oscillatory. I don't know, to me that's actually that kind of makes this whole discussion weird, I think. It
doesn't seem like it's related to neural oscillations.
It's not, yeah, I feel like these two phenomena are separate. It's basically coherence within frequency bands seems to me, at least a priori, potentially completely independent of whether or not signals are oscillating.
Or at least oscillating in any way that we would consider really
that you would see
up and down,
like a peak in the Fourier spectrum thing. It doesn't have to.
Yeah. So in that sense, it's almost a misnomer to call this whole category of thinking like the study of neural oscillations.
Yeah, I think there's at least two things that are being studied: coherence in different frequency bands, like coherence between signals as a function of frequency band, versus the existence of peaks in Fourier spectra of individual signals. And those are often kind of clumped together. In the title of this thing. Maybe not in the title actually, but what does gamma coherence? Blah blah blah. And then the first two words of the abstract are neural oscillations. I don't know. To me, it seems like it's not obvious from the beginning that those are related necessarily.
You need the notion of oscillations to have the frequency, to talk about frequency.
Well, so that's maybe the point. That's not really true. So, so if you have general time series, you can always decompose those into their spectral content. Sure. So they don't need to be oscillating time series for you to be able to look at things like coherence.
Yeah.
If two signals are at all cross-correlated, then at some frequency band you should expect to see some coherence.
Yeah, you could do a Fourier transformation on a spike train, and you'll get a power spectrum from that.
Sure. Yeah.
Yeah, that's related to what you were saying earlier, Nancy. That. Any signal can be decomposed into its Fourier spectrum. Fourier spectrum is sine waves, right? Which are all oscillating themselves, but the whole sum thing doesn't have to contain something
that we
would really perceive as oscillatory.
Yeah.
But it has frequency components anyway.
Yeah, this is just sort of a definitional thing. All time series have frequency components. So maybe we can return to the paper. So what's the point of this paper, kind of succinctly? Would anyone care to summarize?
I mean, I feel like it's just kind of telling people to be cautious because there's these potential sources of like contamination or just the chance to have unclear thinking about how to study gamma oscillations.
So I think that, like, in some way you could view this as kind of a warning / review of the ways to properly think about this, and I think that's good. To me, there's an agenda to it, which is to promote the notion that LFP, specifically gamma rhythms, can be used, and this is kind of a quote, like as an indirect way of gaining insights into neural communication across brain regions, right? And that's related to the title.
You say indirect because it's not spikes?
I say indirect because they said indirect, but yeah.
But also, I mean, it's not just like, if you have spikes, you know that things are communicating. Anytime you just record two signals and they're kind of doing the same thing, you can't just assert that they're definitely influencing each other.
Yeah.
There's always the possibility that they're both being influenced by a third thing and that kind of stuff.
But so they kind of start with, near the beginning, a thought experiment for an experiment that people have actually done. And it's the notion that you could try to look at one network and how it communicates with another network, where from the first network, you record from its output. You record spikes from network A. And network B, you record LFP from near the dendrites, where the spikes would be received by the first network. And so what you would expect to find, if there's some sort of causal connection, is some sort of relationship between the LFP in that second region with the spiking activity in that first region. And they kind of use this as the jumping point, as how this is generally like a reasonable approach, and looking at LFP can be a reasonable thing to do. And I agree with that, and I think that this is kind of a really lucid way of thinking about productive benefits of LFP. But it does strike me that that's not maybe the most representative way that this stuff gets used. Often people record LFP in two different regions, and they want to look at how correlated those are. And so what do you guys think about whether this really does extend well to the issue of recording from multiple LFP signals?
The other thing is not only sometimes people record LFP in both brain regions, or in both networks, if you may, to continue on this example, but also especially in the living animal, there's no way you can target your electrode to the dendrites and not anything else. So then you're really not getting just the field around the dendrites, but the average around the somas and everything around every fiber going through. So it's not just going to be the input to those neurons that are nearby, but also the outputs.
So this paper points out this specific, the fact that where your electrode is matters a lot. And so basically they're claiming that the only time you could expect to really see any high coherence is if you're recording from the spikes of cells in one region, and also you could look at the LFP of a region that first region projects to, but your electrode has to be near the dendrites, which is the location where the inputs come in and hit the cell. And so their claim is that in that situation, if the cells in the first area are spiking with some kind of synchrony, then maybe you could see some coherence between those spikes and the LFP, basically because the claim is that the LFP is just the sum of the inputs to these cells. So if the spiking is synchronized, then the LFP, which is the sum of kind of these inputs from the first area, will also, it will show oscillations and you can find coherence there. But they claim really any other combination of things. If you're recording from cell bodies and cell dendrites in different areas or two different LFPs, like there's just no plausible scenario to get really high coherence from that.
But yeah, that is, as far as I'm concerned, when I read people's discussions of the study of oscillations, and I mean, Nancy, maybe you can comment on this more directly because it seems to me that you're kind of doing something along these lines, recording LFP in multiple brain regions and trying to look at what you can probe from that.
Recording, so in our lab, we've been successful recording single units or spikes in one brain region and then LFPs from anywhere else, up to 6 brain regions. In my particular case, I'm recording spikes in the prefrontal cortex and then LFPs in 2 other brain regions. And I'm trying to ask the question, are the spikes in the PFC being differentially influenced by these 2 LFPs when I manipulate something or behaviorally, etc.? I don't even see that much. I'm curious to know what they consider high coherence. So
I was wondering.
If you, the equation that, the mathematical procedure that Josh was referring to will give you either from anywhere from 0 to 1 coherence of 1 being perfectly identical in phase.
Because it's normalized in the definition.
Right.
Yeah.
So, 0.7, is that high? I look at phase locking rather than coherence, but maybe they're using the term here, coherence, as like more the definition, not the mathematical definition, but like the colloquial definition. It's not extremely high because obviously I'm assaying cells that are not receiving input from the regions that I'm recording LFPs, and also that these cells are receiving like hundreds of inputs. So I see some phase locking, and you can definitely ask if these spikes are significantly modulated by the LFP by looking at circular statistics. So there's ways. So
you're looking at the LFP in the region, and you're saying, are the spikes in
another brain region that receives anatomical projection? So that's an important.
So you're also going in what seems to me the opposite direction from what this paper is talking about, right?
That is true.
So how do you guys think about that? And here they kind of have an anatomical model.
Right, so it's a little bit of a circular argument, like, well, whatever inputs to the brain region we're recording to the LFP are definitely affecting the output, which is then affecting the spikes over there. Maybe if we had a perfect model and we could like only record from the dendrite, then we will see that the high coherence will only be in the scenario that they're describing. But because our LFP is not targeted to the dendrites, we're really getting contributing spikes of that brain region, which is the output contributing to the LFP as well.
So that's a positive in your case, though.
Definitely.
Yeah, and so for some people that's a negative because they're trying to study the input to an area or something like that. But it could be helpful if you're actually just trying to get some measure of the overall activity.
Right.
So this paper claims that there's just current limitations in what you can record, and I think you would agree with this, Nancy. Like, if you could just record all of the cells in both areas, that would be lovely. They understand why people use LFP, but, if you really want to say anything strong, you need to be recording from spikes along with LIP, or just have more concrete knowledge about the connectivity, like the actual connectivity, to be able to say anything. Like, if you had no idea the connections between 2 areas, you just picked 2 areas at random and tried to look at their LFPs and use their LFPs to determine if they're connected, that wouldn't be sufficient because it's too weak of a signal, it's too weak of a statement. But if you have a lot of other knowledge surrounding the circuit that you're studying, then, you can use LFP on top of that to say things.
Specifically anatomy.
Yeah, so some of the core points of this paper are that it's important to know in which layer the LFP originated. They view that explicitly, and my understanding of the work that comes out of Braaaiiiiinnwaaaaaavess's lab is that they actually use depth probes with many electrode contacts on them so that they can try to actually localize the LFP that they're recording and try to figure out which layer it's coming from.
Right,
so that's a special choice that they've made that gives them more power.
Because they're very interested in the different layers and the sources of these LFPs. And so these chunks that Josh is describing, their electrodes have what we know as high impedance, which means that the signal that's recorded by that electrode is very, very local. The electrodes I use, because we're interested in the sum activity of that brain region, have very low impedance. So then they're really going to go across layers.
So it's almost misleading to think you guys are recording the same signal. Right?
Absolutely,
all of this is called LFP.
It's funny, yeah.
Yeah, you're recording something that, do you kind of have an approximation for how much volume you're recording from? Like how many neurons you think you're getting in your LFP signal?
I will be making it up, really, because I don't...
Okay, but you believe it's a large region.
It's definitely large.
Whereas here, they think that they can localize it to the specific layer.
I've never tried to record with chunks and observe across electrodes to see just how different it looks.
Sure. But there are some kind of bigger issues, I think, that we can now start to talk about when we talk about neural oscillations and what kinds of things you can extract from them. And something that I think about is that it strikes me as extremely difficult to really understand mechanistically what's going on when you record from this stuff, when you record LFP in general. And so here, with this kind of good anatomical model, if you think, hey, I'm recording from a specific region, and I'm also recording from another brain region, but in the first region I know where my LFP is coming from, or in the second region I know where my LFP is coming from, maybe you can make some principled hypotheses, but I don't know if I believe that there's, and I'm open to being persuaded, I don't know if I believe there's really that much that you can learn from recording these kind of very gross signals, very, very crude, like
when you say that
signals.
much, you mean mechanistically of how the brain is working?
Yeah, I mean, if I wanted to build a model that performed the computation that I think a given brain region is performing, what could I pull from just oscillations? So, right, there are special cases where you record from a whole bunch of single units, you get lots of spikes, and you also record from like some LFP signal, like in the hippocampus, where people have done some interesting things looking at how specific spikes are related to the phase within an oscillation.
Right.
And I think people with a kind of computational bent are sympathetic to those kinds of approaches.
Yeah, but that's really only one example.
Yeah, that's an exception, kind of. Most of the studies of oscillations, you record at multiple LFPs, and you're really not looking at spikes very much. Right, again, with exceptions, but and people are looking at kind of coherence between these different brain regions with respect to the LFPs. You yourself are doing this. I want someone to tell me kind of how that could inform models of how the brain is performing the computations, because that's the thing that I think I'm most skeptical of when I hear about studies using oscillations.
I guess that would require a person that knows about models and about LFPs as well. So,
I mean, that's a pretty general question, right? Because, like we said, what we call LFP is some very aggregate signal that will change depending on the properties of the electrode you're using, whether or not you're talking about where the electrode is located, say, within layers of, cortex or something like that. So, I mean, there's like multiple specific questions you could ask, right? Like, there are specific hypotheses that exist about areas coming in or out of certain types of coherence and how kind of pseudo-mechanistically that could influence things like spiking behavior and so on. And then there's also pragmatic questions, right? Nancy, you kind of alluded to some of the pragmatic benefits of recording, things like LFP earlier. You can ask specific questions like, if I record this specific signal, so some specific LFP, I use this kind of electrode in this kind of brain area, we could try to figure out how much that can tell us, say, about spiking in that region, for example. Like, that would be kind of a pragmatic thing you could do, which is kind of different to what a lot of people talk about when they talk about oscillations. But, like, I don't know, I think you can break your question into kind of many different questions about. There are several kind of things you could think certain LFP signals could help you with pragmatically, just easier ways of getting at whatever.
Is this brain region active, yes or no?
Things like that, yeah.
During this.
Versus like more
knowing
specific.
How the brain is working. Well, but going back to the models, sometimes when I think of LFP, I think of rhythmic activity. And let's just forget for a moment of like the going up and down. But like, I feel like if you are recording LFP, you can see these different frequency activities that are occurring in that brain region. And I feel like if you were building a model and you know that this brain region A is active in these frequencies while the brain region B, which is connected with that one, is acting in these other brain regions. Frequency, sorry. I feel like that could help us understand how the brain is working. That frequency matters.
So I feel like that intuition is at root for a lot of people who study oscillations. But I think for a lot of modeling people, it's there's a recognition that it's very easy to produce oscillations in networks.
It's kind of it takes a while to not produce them.
Right. Yeah,
It's kind of the default.
Yeah, so there is an interesting thing, right? In theoretical neuroscience, one thing that people have tried to do is to build large populations of neurons and wire them together in ways
and they oscillate naturally?
Yeah, wire them together in ways to see like what kinds of regimes of activity
and there's plenty of like parameter space where they will oscillate.
Do they have preferred frequencies? Or do they oscillate
there's lots of studies that have been done on this. I don't want to
I mean, I don't know. How easy is it? Like, just a simple thing. Record LFP in Cortex, get the power spectrum, build me a integrated fire neuron network that has the same power spectrum. Is that easy? I don't know if that's easy.
They're saying that their networks naturally
you can get things that oscillate. You can trivially get things that oscillate like
but
in ridiculous ways that are
frequencies.
not brainish at
So
all.
you're saying,
Yeah, I'm not sure how well it relates to biology.
Well, yeah, there's a sense that there's something productive about the oscillations.
Oh, well, I was going to say that also, if you give, someone who's trying to build a model based on individual neurons, build like a computational neural network, and all you give them is the power spectrum, like that's incredibly difficult. There's really nothing to go off of there. And so I think that's another reason why it's not as clear what role they play, because the relationship to the individual cells is so tenuous, and probably there's many different neural circuits that could create the same LFP patterns.
I've actually read a couple of reviews that claim that the spikes are a very minimal contribution to the local field potential, that the majority of the amplitude of the field comes from subthreshold activity. And most of these networks, I know very little, do they have subthreshold activity that occurs?
So when people make artificial neural networks that are supposed to be kind of biologically plausible, they usually represent them as having, like a voltage that when it accumulates to a certain threshold, the neuron spikes, communicates with its, the neurons that it's wired to in this simulation, and then it resets its voltage to a low value, and as it receives inputs from other neurons in the network, its voltage increases again until it spikes again, and so on. And that's basically the model for some number of neurons. Okay,
so then there's not probably no models taking into account just the currents that are constantly going.
Well, no, so it's receiving currents from the neurons that project to it in the simulation, the same way, roughly.
There are people who are trying to model aspects of oscillations and that sort of thing, and so you can explicitly make sure that your neural model includes enough details that you feel comfortable that you could say, I can, estimate what the LFP in this network would be in this model network.
Yeah, I see. But going back to Josh's point, because the local field potential has so many multiple sources and it's unclear what the distribution of those sources are, and if that depends on brain region, which probably does, and depends maybe on brain state as well, and behavioral state, we're still at the level that LFPs are not going to inform models of the brain. Maybe.
I want to be clear that maybe,
perhaps.
I mean, if you're recording a small number of channels of LFP, there are exceptions. If you're recording large numbers of LFP signals across many channels, I've seen work that can do interesting things with that, and I'm not trying to ubiquitously criticize this, right? It's not clear to me from looking at these kinds of signals, unless you want to build a model that's kind of in terms of these abstract variables, right?
Trying to explain the LFP.
Exactly,
perhaps.
right? So,
that's what we think we need, actually. People that can model and say, how can this happen? How can these frequencies occur?
In terms of computational neuroscientists, I think they think of the building blocks of the circuits they want to build as neurons. And so it would be a very kind of weird shift. And I think maybe part of the disconnect in language comes from the notion that, in order for a computational neuroscientist who's building, models of neurons, to build a model that is going to make useful use of data that's predominantly kind of LFP oscillation style data, it would have to model these kind of abstract variables.
I think that's really interesting, though. I have to say, I felt very ignorant when I read this, because it really made stark to me reading these papers, and especially the one which we haven't mentioned, which maybe we'll touch on briefly, this other paper about ephaptic coupling or whatever. It made stark to me this idea that, if I think of a network of a bunch of, say, a model network, like a simulation of, say, a bunch of pretty, what I would consider kind of detailed neuron models, right? Say, some model of a neuron that actually takes into account specific membrane currents of various different types and so on, and I link them all up together. I'm kind of thinking, that's a real detailed type thing to be studying, and it actually isn't the kind of thing I would be most interested in studying, because I want simpler things so I can focus on computations. But if you were studying something like that, I would be thinking that's very detailed. And the fact that neurons are embedded in this complicated soup, they're all very close to each other. Yeah, it's highly conductive.
The physical aspect of the neurons is sometimes lost.
I think that's really interesting because, in a model, they're only touching each other via synapses, and you have total control over those synapses, right? And that is really distant from the reality of it. I don't know. I do find it kind of interesting. I'm not sure,
I think it's on the same level as astrocytes and glial cells. We just sort of ignore them. Those as well have.
Most of the cells in the brain are not neurons, right?
Right. Yeah. We're very much action potential centered.
Yeah. Although there are examples of computational models of neurons, neurons being the unit of the model, that use LFP to constrain facts about them. But the problem with that is that you have to, as a modeler, come up with what the LFP is. And so you kind of just take a sampling of the inputs and kind of, filter it in a way and say okay, that's probably what the LFP is. But without knowing biologically the source of the LFP, it's very difficult to try to model it. I mean, you're just making up a definition for it.
Yeah, I actually have to say, when I was thinking about this, I was like, this would be so stupid, in some sense, as in people who want to know how the brain computes wouldn't give a shit. But I actually was thinking, it would be kind of cool to build a model of physics of brain tissue. I mean, people must do this. I don't know. Just actually think about how homogeneous is the medium, blah blah, all that crap.
How much voltage can it conduct or current?
Yeah.
People do it, but we just don't care.
I think it's kind of cool actually in a way, but I wouldn't. I don't know. I mean,
I think people have studied this because they do simulations about how far you can record from neurons.
It's probably hard, right?
Yeah, not super accurate models.
Yeah.
It's also used. It doesn't feel like it's computational neuroscience because I think it's people who just want to understand their tools better. It's we'll just run some simulations to know what we can say about this. It's not lumped in with the rest of computational neuroscience where you're really trying to understand the brain.
So one other thing that I wanted to talk about, though, because it's related to the points that we've been bringing up with respect to oscillations, is I kind of feel like there's almost a bait and switch that goes on. Kind of in oscillations in general, I feel like you say, hey, what's an LFP? And someone can give you a very concrete example in one setting, but then in other experiments, they just kind of they're not as precise. So at the beginning, they start with a very concrete, very lucid, realistic setting where they tell us, here you can understand what an LFP is, and so let's trust this. But then by the end of the paper, they're really talking about some high-level stuff, and kind of something near the end says that, we hope with a full understanding of their tools, neuroscientists will soon be able to decipher the precise mechanisms and roles of oscillations, synchrony, and interregional coordination in carrying out the sophisticated neural operations underlying cognition and behavior. So it's this kind of grandiose thing where oscillations are sort of the causal mechanism. Right? They've started by kind of asserting that we're using LFPs to record something that we know what it is, the inputs to this dendritic network. But then at the end, they've just totally switched, and now they mean something more grandiose by the use of LFP. And it was a gradual thing in the paper.
I think most research articles do that at the end, and they make a big claim, by this type we're going to cure something. So
I feel
really
this claim at the end, in particular, is unrelated to the way they sold LFP is something that I wanted to get behind as this is just an indirect mesoscopic measure of dendritic activity, and we know which layer it's from. To at the end, we're going to record multiple LFPs, and they're causally interacting, and cognition is emerging from oscillations. That's the kind of use of oscillations that I'm very uncomfortable with.
The end of it does end up being more in line with a lot of research that has come before this paper, which does, want to say, a previous Bujaki paper even hints that, oscillations might explain consciousness. And,
that's the kind that I'm most skeptical of, because it seems pseudoscientific to start with something very consistent, rigorous, logically well thought through, and then just kind of make it jump somewhere. That it's not even clear why oscillations have anything to do with cognition at a high level.
There's a bunch of papers of monkey behavior showing that when they're paying attention and behaving well, you have a lot of gamma.
Sure, but it isn't the way that they talked about LFP at the beginning of the paper when they presented it.
It's very different. Yes,
it's very different than anything that you would jump to talking about cognition.
Why is there one term for this?
That's kind of the point.
Why are we always talking about LFP? It's insane. Just to be clear, LFP usually is you take your signal and you filter it brutally below some frequency threshold. That's could be, and we've said this a million times.
Maybe it's saying voltage. Okay, it's just the signal. It's as
membrane voltage is more of a neuron. Is more specific.
No. I'm not saying membrane voltage. I'm just saying a voltage.
Yeah, it's just voltage. But it's LFP. Given that, it should kind of never end up being spoken about as a cohesive item.
Sure.
You should have to talk about some...
The LFP of blah, blah,
Yeah,
blah.
I'm recording the broadband signal in the dendrites.
Yeah, and then you
just call it the broadband signal in the dendrites.
Mm-hmm.
Or I'm recording...
The low...
low
pass filtered frequency that for like a huge volume of the brain.
This much volume, yeah.
And people kind of pretend and seem to think that there's a strong relationship between like recording an LFP and looking at an EEG signal. Mm-hmm. It's not clear to me that we should think of those as the same kind of thing or expect to see the same kind of information in it. Mm-hmm. But kind of as one final push, and I want to hear thoughts on all of these points. What is it that determines what kind of models or thinking about signals people are comfortable with? Right. So why is it that some people would feel comfortable looking for information flow or like directed information in these signals and expect to learn something from it? Like I'm going to look at one brain region, another brain region, same way that people would in fMRI science. They talk about a network of 4 interacting brain regions and think that they're mechanistically understanding the brain. Whereas for other neuroscientists, they would say, well, that's not giving me any information. Does anyone have any thoughts on why?
Yeah, this is a thing that I've always struggled with oscillation type things because of, I can agree, like, okay, you find these things that correlate with attention or with whatever other behavior, but then I try to envision what's actually happening to the neurons and how that's information being processed and passed along in a way that fits in with everything else I believe about neurons and how they function. And then I kind of hit a wall, which is like, oh, wait. Especially this like kind of causal loop, because people talk about, LFP, if you're in a up or down state with the LFP, that'll determine if the cells will be more excited. Well, it's both ways. They say the cells are more excited, and so if they get an input, they'll spike. But at the same time, LFP is supposed to be measuring the input, so you're just saying if they get more inputs, when they get inputs, they'll spike or something like that.
It's extremely circular because LFPs are partially contribution. The contribution is like the actual spiking, so...
Yeah.
There's no such thing as an LFP. We should never talk about LFP like it's a thing.
Well, I don't just mean this for LFP. I mean,
it's the field that whatever electrical signal you get outside
from
the
a
cell.
recording tool, there's an LFP, right? It's whatever.
It's just not helpful to just call everything that you pick up with an electrode that isn't like placed intentionally near a cell an LFP.
What do we mean by a thing, right? It's like kind of somehow you would mean you would have some very specific model in mind of how certain signals arise, and you'll be able to predict things about those signals.
Okay, so you're saying
this kind of gets to my bigger point, which we can almost phrase as a modeling point. Again, I mean, if you see 2 brain regions or 3 brain regions or 4 brain regions, and you treat each of those as having like one variable or maybe 2 variables that are interacting, what does it mean for you to say that you've learned how the brain works by looking at 2 variables from different brain regions interacting, or a small number of variables from each brain region interacting, right? I say this brain region influences this brain region, so I know how the brain works.
In this way. No, who say that?
No, I don't mean that like they would jump to that extreme conclusion, but there's a sense like I've gained meaningful insights about how the brain works by saying that this brain region influences this brain region. And seems very...
How far is that from lesioning brain regions and saying, look, I learned?
So I would say it's not very far.
This is a step forward.
It's a step forward, but like,
because
it's a small step forward.
It gives you temporal information, and it gives you...
It's a step sideways.
Yeah.
It's not always
So
I actually have a problem as well. Like, it is true, it's very easy to find problems with recording these signals, let's call them local field potentials, and feeling like we've learned something about the brain. But even though spikes are very precise and they're a thing because we can say exactly how they happen, I also have a problem with myself recording only 100 neurons and thinking I can actually say this is happening in this brain region when this brain region has thousands and maybe hundreds of thousands of cells. So
so that's problematic too. Yeah,
there's problems everywhere.
So an electrophysiologist is going to record what they believe to be a representative sample of the neurons in a brain region and try to infer about what's going on in that brain region based on that sample that they believe.
So can't we infer from the local field potential as well then?
Yeah, we can. Yeah. But it's like funny. I mean, I think it's a good point, right? Like people people got very excited at some point when they realized, oh my god, this one neuron correlates with behavior, right? And they were like, holy shit, that's insane, right? And then this became this like kind of thing, like the spikes of like single neurons tell you things and so on. And that kind of dominates in a way this idea of like representation, like the spiking rates of neurons represent things. Neurons have receptive fields. How much that neuron is spiking represents something that's happening in the world and so on. And that's lovely and everything.
And the same happens with frequencies and oscillations, I think.
Yeah.
Well, gamma increases when this thing. Gamma represents this thing.
For me, at least, there's kind of like a satisfaction when I see, this is when a neuron is spiking, because I know, okay, that is when it is sending a signal to the neurons that it is connected.
Yes,
It's
exactly.
releasing neurotransmitters.
Yeah. And so, like, I can at least say, okay, this is communication happening. And maybe to really understand how the communication happening is what it's doing, you need a lot more cells and more information about connectivity. But at least I can say,
it's the right class of
So that's fine, and that's nice, right? And that means that one of the reasons why that's so satisfying is because we have a model in mind. We have a mechanism. We know how a cell spiking a bunch when there's a thing moving can influence other cells. Other cells can know about that via the release of neurotransmitters at synapses. So that's great.
And specifically, right, it's a sufficient explanation that neurons produce behavior, right? We know that neurons influence muscles directly. Mm-hmm. So it is very direct, right? If we know that neurons are producing some activity.
I mean, it's sufficient. It's not sufficient. I mean, it's sufficient for some things, right? It's sufficient for explaining how a neuron and motor cortex can make a muscle move. But, like, I think this gets to what some of these people try to talk about, these oscillations folks, right? There's loads of things that we can't explain. I think a lot of them haven't been framed very coherently, and that's part of the frustration. But the notion is that somehow quick coordination of communication between brain areas will probably be important for cognition, and like maybe oscillations have to do that. Yeah. How can you explain that just with spikes? You probably can, but we don't have models that actually explain that. So it's, we don't have sufficient like frameworks and explanations for like a bunch of things, and people are kind of trying to get at them. Yeah. And I think like the spiking thing, it's possible. Like the gamma coherence, whatever, like one simple model might be one region is fluctuating and another region is fluctuating in phase via whatever mechanism. And suppose it's the case that when that's happening, spikes that are being sent from here to here are more likely to make region B spike, right, or something. Then that could be a mechanism by which, if you can modulate the phase difference between the two areas via some other unknown mechanism, it could be a way for controlling communication. This is like an idea, right? But like it has millions of holes in it, right?
Yeah,
but I mean, there is the possibility.
They call this idea has a name. It's like communication through coherence model.
Right.
This is what they call it.
And this, I mean, that is at least a possibility of something analogous to the kind of spike neurotransmitter spike thing, right? But it's just we're so far from seeing that actually happen.
We're far from a mechanism and like a model being able to explain how is that possible.
Yeah, and being able to like show it the way we kind of see. We know that spikes fire we can measure all these things. Neurotransmitter releases, it increases or decreases the potential of the next cell.
So I think maybe something that I've kind of been thinking about as you're saying these things, and something that's influenced by the way I was thinking about this before. To me, it's almost as if the reason why studying oscillations as the phenomena is somewhat unsatisfying is because I don't feel like a model that just relied on oscillations would be sufficient to explain the computation I believe the brain to be performing.
Yeah, but maybe a model that just relied on spikes wouldn't be sufficient.
Well, so maybe it wouldn't, but I believe at least that neurons are the kind of largest unit that still is small enough to explain the computation, right? Like a whole brain region is larger than a neuron, but it's not small enough, right? There's too few brain regions for me to believe that just if you describe how the brain regions interact, you can explain the computation. But in terms of neurons, that's the largest unit, that if we treat those as the units, we believe we could build a model that could actually do something. So like artificial neural networks, which are used to perform computations, the unit of those is a neuron, or a neuron-like abstraction. We believe that by making a model that's in terms of hundreds, thousands, tens of thousands of neurons, we can actually perform a computation. If we build a model that's in terms of abstract variables that correspond to like oscillatory activity in a few brain regions, we can't make that do anything. So as a person who wants to like build kind of models of brains that can perform the task that brains perform, it isn't clear what I get from adding the oscillations. Now, maybe
I think it's very open that
No, it might help. So maybe after I have a model in terms of neurons, I can make the model better by adding some notion of oscillations to it.
So I think this would be a good time to talk about the second paper briefly, because it does give some idea of what a mechanism of LFP is that could actually influence spiking of cells, and then that, brings everything together such that we would, if the second paper is true, then maybe you would build a spiking network and it would be lacking something, and LFP would give you that thing that it's lacking. So that paper is called Ephaptic Coupling
of cortical neurons. Yeah. This paper is by Henry Markram.
And so this term ephaptic is basically referring to this notion of the LFP that surrounds a cell is influencing that cell. So we think of LFP as being some measure of the inputs to cells in a region, but also it's floating around in there with the cells, so it could be influencing the cells itself. And this paper was trying to address that. And so just very briefly, how they go about that is they have cells in vitro, so in a dish, and they block all of the neurotransmitter signals in these cells so that there's no signal that's coming onto these cells from other cells so that they can control all the signal that's happening. And then they place electrodes in regions around the cells to kind of induce their own LFP that they can control. And then they can measure from inside of the cells if the cell membrane is responding to this external signal that's supposed to be like an LFP. And importantly, they keep the external signal around the same magnitude that a real LFP would be. Because obviously, if you put if you stimulate a brain region excessively, you'll of course influence the cells. But the question is, can LFP be influencing the cells?
So particularly, they're leaving it at subthreshold stimulation that would not make the neuron spike.
Yeah. And so what they show is that indeed the membrane potentials of the cells that are near where they're stimulating, they have oscillations that they claim follow the LFP that they're creating. And then also if they stimulate the cells so that they're spiking and then also have this external signal, the spikes will become more in line with the phase of the external signal than they would be if there was no external signal there. So they're claiming that this LFP that they've created, which is supposed to be like a real LFP, it doesn't change the number of spikes that a cell would have, but it changes their precise timing. And so you could argue that for some encoding of information, precise timing matters, and so the fact that LFP can make groups of neurons have more synchronized spikes could be relevant.
So maybe I need ephaptic coupling. Maybe I don't need ephaptic coupling. But sure. we kind of know we need the neurons.
Yeah,
Studying the kind of higher level of resolution, I don't know, it's still invasive. It's not clear to me why people do studies that are just in terms of LFP when they could be recording spikes. So maybe this is directly a question that Nancy can help us with. I mean, you're already doing an invasive study. It's not like you're saying, these are humans, I can only record like outside the skull.
I mean, it's still sometimes hard to record spikes, for one thing.
Yeah, that's why I was the only reason somebody will record global field potentials and not spikes, that means that they have all the hardware that they need to record spikes. It's just that it's very difficult to record single units. It's much more delicate. So during the surgery, if I accidentally touch with some, particle, some dust, if there's some dust on the tip of my electrode, the impedance of the electrode for getting single units, because you want the signal to be so localized, so then you're isolating one cell, the impedance is so high then that anything could disturb it, and then you get zero signal to it. Now, with the local field potential, the animal, during the surgery, you could have blood, you could have a lot of things, and still you'll get a signal. So it's just simplicity. But that obviously comes with a lot of, it's simple to get the information, but how can you interpret it if you don't have any single unit activity, which is what we know the mechanism of. But Josh, I think that you're being very close-minded to think that the local field potentials wouldn't add anything to your model. It's possible that you
say
cannot make it add anything to your model because you don't understand it enough, or like there's just no precedent. Like, how will you start with that?
I think that this paper about the ephaptic coupling is a good example, and it's also kind of related to things that modelers already think of. Like if you model something that you kind of call a rate unit in a model, you might actually think, well, I'm calling this kind of one unit in my model, but in reality it might be like 100 cells and their average output is this rate. Well, maybe those 100 cells are coordinated to have synchronous spiking so that they really give you like a strong output signal that would really drive a downstream cell. They're coordinated that way by the LFP. I'm not saying that this is necessarily the case. I just feel like there is a conceptual mechanism that could be worked in if it really is the case that LFP is controlling spike timing. But it might be controlling spike timing. It might not be controlling at all because this is not an in vivo experiment.
Right.
And it might be controlling it.
It also doesn't show that there's a role, even if hypothetically it could.
Yeah, no. But it could be, controlling it so subtly that it doesn't really matter. It doesn't matter that those cells are spiking 1 millisecond.
Right. Timing matters maybe for some things, but not for others.
Yeah.
So.
But I still think that there's a framework to incorporate these kinds of things.
Yeah, I wasn't saying that. To me, I'm still skeptical that when you look at studies that look exclusively at LFP kind of signals, that there's much that I can do with that.
Yeah, it's definitely the case that everything needs to be taken into account. And just like it's hard to interpret the data without some notion of the anatomy and what exactly you're recording, it's hard to build a model based on LFP if you don't know anything about the anatomy of that area or you don't have any single cell information.
It's only the papers where the people have like really high and rigorous standards that are actually that useful anyway.
I feel like if you've honestly recorded from lots of neurons in a given region, it's not clear to me how that data is totally not valid. If you are honestly reporting what you did while those neurons have been recorded, I feel like there's some use you could get from it. People can look at your neurons, they know what they're getting, and they can look at the task or the paradigm that you're using when recording from those neurons, and kind of know what those neurons are related to some extent. And I mean, maybe this just means I'm thinking too much in a way that's consistent with the way that people think about neuroscience today.
You're very dogmatic.
Well, yeah, but there's a kind of weird alternative dogma around neural oscillations.
Well, I don't think it's an alternative. I think it goes together. It's an add-on.
I think, to be fair, for some people it's an alternative. So maybe not for you, Nancy, but there are people who kind of really praise oscillations. Like, oh, it's the mesoscale explanation we've all been waiting for. It's like, we can't know about all the neurons, but the whole brain is too much, and now we found some middle area that will obviously be helpful.
This is always a problem in neuroscience, though. It's like, who are you talking about?
Yeah.
Are you talking about the crazy people, or are you talking about the 2% of reasonable people? And within the 2% of reasonable people, most ideas are still maybe valid because nothing is that constrained, right?
Yeah.
It's always like that. It's like a scale-free subject.
It could be anything.
Anything can be crackpot, and anything can be totally relevant.
Okay, well, I think I want to thank Nancy for joining us today.
Yeah, definitely.
It was really nice.
Withstanding Joshua's assault.
Thank you for inviting me.
Okay. Till next time.